So we'll continue with the presentation on regulation of gene expression in prokaryotes. So we have seen that simply an operon consists of structural genes and regulatory genes. So there are regulatory proteins which interact with the regulatory sites. The regulatory proteins can interact with the DNA, that means with the regulatory sequences, and this binding will convey a signal, and this signal will be either a positive signal or a negative signal. And based on this concept, we have two types of regulatory mechanisms. One is positive regulation, other one is negative regulation. So we'll see in brief what is a positive regulation and what is a negative regulation. So the bound protein, that means the regulatory protein, either facilitate or inhibit transcription. That is the two possibilities, right? So in both the cases, the signal will communicate the system, right? That means usually in case of a positive regulation, the binding of certain molecules will provide a positive signal. At the same time, in negative regulation, the binding of certain molecules to the or certain proteins to the DNA will cause a negative signal, right? So that is the simple explanation. In both cases, the regulatory molecules will interact with the DNA, and if it is a positive regulation, or we count this as a positive regulation, if the binding of the protein to the DNA induces transcription, or which promote the transcription or enhance the rate of transcription. At the same time, if binding of a regulatory protein to the regulatory site reduces the expression rate, then, or which prevent the expression or prevent the transcription, then we count this as a negative regulation. So you can have a lot of explanation for each class. We'll be seeing in this briefly, as already we have seen the lac operon mechanism in detail, and that detailed explanation is representing the negative regulation. Actually, here the repressor, there is a repressor protein which is actually synthesized by the lac I gene, that means the repressor gene. This repressor protein in the active state, this active repressor can bind onto the operator site, and this binding of the repressor to the operator site will prevent transcription of the structural genes. So the binding of the repressor to the operator will block transcription. So this is a kind of negative regulation, right? Then this condition will be reverted, as already we have seen, by the presence of inducer which interact with the repressor, and this repressor will be removed, right? And you can also see, so lac operon is a classic example for negative regulation. Not only lac operon, even the tryptophan operon or the tripoperon is also regulated by a negative regulation. And in case of tripoperon, the repressor actually synthesized in the inactive state, and this inactive repressor is activated by interaction with a co-repressor called tryptophan. So this tryptophan repressor complex, that means now this is an active repressor, this repressor will bind to the operator, and this will prevent transcription of structural genes in the tripoperon. So in both cases, binding of repressor causes decrease or which inhibit transcription of structural genes. So this is what the negative regulation is. There is the promoter which is bound by the repressor protein, the operator bound by the repressor protein which prevent transcription, and if there is no repressor, there will be transcription. So this is what is seen in lac operon and tripoperon. In both cases, the repressor bind onto the operator site, and in case of lac operon, the operator is bound by an activated repressor, and this active repressor is synthesized by the lac I gene. But in case of the tripoperon, the synthesized repressor is in the inactive state, and they are activated by the apo-repressor or the co-repressor. So again, all these images will give you much more idea about the lack operon and the negative regulation. This is the image shows you the regulation of tryptophan operon under negative regulation. You can see the structural genes are synthesized only when there is no repressor bound onto the operator site. Now about the positive gene regulation. Usually the binding of these proteins, binding of the regulatory proteins to the DNA will enhance the rate of transcription. Usually these are usually what we categorized as the activators or enhancers. So these molecules will greatly enhance the rate of transcription. Usually in bacterial system there is not there we cannot see much elaborated activators or enhancers, but rather in eukaryotes you can see various examples for activators and enhancers. But still in bacteria we have certain positive regulatory mechanism that we will see. Okay, so the basic concept is like the activator is initially active and is bound to the regulatory site and enhance transcription, and this causes usually there will be a positive regulation. That means there is a positive signal coming due to the binding of this particular protein. And in case of positive regulation also, at certain times just like we have seen in the negative regulation, the binding of this molecule sometimes the removal of certain molecules will cause activation. That means the activator is initially in the inactive stage and gets activated on binding with certain signals. That will be the condition in certain cases, and this activator molecule can enhance transcription, right? So we will have a detailed explanation with this lack operon mechanism for positive regulation. So this is the positive regulation in lack operon is usually denoted as the catabolite repression, or catabolite repression is the positive regulation for lack operon. Here the activator molecule is called as CAP, or catabolite activator protein, and here the effector signal is the CAMP molecule, right? So the concentration of CAMP, why the CAMP act as an effector? Because the concentration of CAMP is inversely proportional to the concentration of glucose, and we know that when glucose is present, the operon has to be shut off, and when glucose is absent or less available, then the system has to be on. Okay, so the CAMP and CAP molecule will work together for an increased rate of transcription of lac operon genes. So the CAP usually will be in the inactive state. CAP is nothing but a protein which can interact with a binding site in the which can bind to a regulatory site in the lac operon, and this binding site is usually denoted as CAP binding site, or CAPB, CAP or catabolite activated protein binding site, and this is usually located in or near the promoter site. So this CAP will be in the inactive state and they becomes activated on binding with CAMP. Okay, so once CAMP is bound onto CAP, which is active now, and this CAMP complex can bind onto the CAP binding site in the DNA molecule, and this binding causes an increased rate of transcription for the structural genes. So usually this positive regulation occurs along with the negative regulation. And here you can see the binding sites. The CRP site is provided there. It is usually near the promoter site. This will give you an idea. So here you can see the inactive CAP, catabolite activator protein, and the CAP is activated by CAMP, and the CAP complex will bind onto the CAP binding site, and now that causes increased rate of transcription. And at the same time, this operon is already under negative regulation, and these operons rate of transcription will be enhanced by the catabolite repression. So all these images will give you idea about how this CAP complex will help in the regulation of lac operon. And one more point here we have to remember is about the concentration of CAMP. We know that CAMP concentration, it will be connected to the concentration of glucose, as already stated. That means the CAMP will be available in the system only when glucose is absent in the system. This is because when when glucose is available, most of the CAMP will be converted into ATP. That means most of the AMP will be converted into ATP. That means the concentration of AMP will be less and concentration of ATP will be high. So the condition is like there will be CAMP molecules. Usually CAMP molecules are nothing but the cyclic form of AMP. The CAMP will be converted into AMP when glucose is present, and this AMP will be converted into ADP, and from ADP usually the ATP will be generated. So all these reactions occurs when glucose is available in the system. Okay, so when glucose is present, normally we can imagine that the concentration of so when glucose is high, see that when glucose is high, normally the CAMP will be less. So that will be the condition, or that is the relation between the concentration of glucose and CAMP. Similarly, when there is no glucose available, then usually the ATP available, most of the available ATP will be converted into ADP. That means that will be consumed, and this ADP will be further converted into AMP, and this AMP will be cyclized into CAMP. So this will be the condition when glucose is less available in the system, and that means when glucose is less, usually the concentration of CAMP will be high. So this is the correlation of concentration of CAMP and glucose. So what's our point is that the CAMP concentration will be high when glucose is less, and this CAMP will activate the CAP. So the CAP once activated can bind onto the promoter site, and this will cause expression of the structural genes of lac operon, and which will consume or which will break down the lactose into glucose, and there will be release of energy. That means the release of ATP. So again the concentration of CMP will come down and concentration of ATP will rise up, and there will be reversal of this process. So this is about the positive regulation in lac operon.